So I'm Paterne Agre. I'm a molecular breeder and then a bioinformatics scientist based at IIT. And today I will be sharing my work related to yam and then how we are using different genomics information to accelerate the in yam breeding. So let me share my screen. Okay, so can you see my screen? Brandon, you just give me the signal if this is
Yes, this looks good.
oh,
Thank you. Please go
okay,
ahead.
perfect. Yeah, so integrating genomics and pangenomics in yam breeding pipeline, and then I'm presenting this one on behalf of the yam team, which is kind of like a lot of people. So
Oh,
I will focus on
I was
just
finally able to join again. Hello, can you hear me?
yeah, Carlos, we can hear you.
Yes, yes. Well, I apologize to the audience, but it looks like something happened to our Zoom connection here in NC State. Welcome, everybody. I don't want to take any time from you, Agre, but we are pleased to have you present this time. And please, people, tell us from where you're joining. And go ahead, Agre.
Okay, so thank you, Carlos. So I was saying that I will just follow a common present outline, a brief introduction, genomic resources we have so far for the YAM, our marker association, validation process, the pangenome we have, and then also talk about the functional genomics and the annotation we have done so far, and then future development. So in yam, we have a lot of different yam species in Africa. It's totally different from other crop whereby we have more than 600 species, out of which 11 are cultivated. And then the total production across the globe is 89 metric ton, and then we have almost 95% of the total production is from Africa. And then over 400 million of people depend of this crop. And then you can see in terms of kind of like the value of production, you can see the market value of the market value of yam is equivalence of all other roots and tuber crop combined, which is a very, very huge potentiality. In terms of challenges related to yam production, we have some disease impacts, which can lead generally up to kind of like 100% yield loss, among which we have the yam mosaic virus, and then we have the yam anthracnose disease. We have also some post-harvest loss also, which is also is very, very damages, and then we can attribute this one to the dry roots and then to the dry rot and then to the wet rot, which are related also to the nematode. Another point also that is kind of like very important to raise here is the staking aspect of yam, which costs almost 30% of the total production of the yam. In the yam, we have different species, which is also kind of like another challenges for the breeding program, whereby you cannot make an easy crossing within the same yam species and then among some species due to some ploidy differentiation. We have the flower issue. Some of the variety that have good attributes, they don't flower, which means you cannot use them for breeding purpose. The breeding cycle is very lengthy. One full year we cannot kind of like do 2 year, two cropping season at this for the same year, which is kind of like a very challenges for the young breeding. But we have our breeding objective, which is not very different from the common breeding for other crop, where we are focusing on kind of like high impactful variety of young with key attribute traits. Among the traits, we have focusing on high yield and stable tuber yield, resistance to biotic and abiotic stress, and then we are focusing on better food quality, which is very important, especially for yam, which people are looking for the tuber quality attribute. So to do this one, we have defined very well what are we breeding for. And then if you remember very well, when I started this presentation, I said that we have a lot of yam species, but we are not working at IIT on all those varieties. 11 are cultivated, but within IIT, we are focusing on two. What we have, the white guinea yam, and then the water yam, what we call alata. And then within the white guinea yam, we have two breakthrough products, or the quick bushy. When then here we are focusing on having a semi-dwarf yam, which we not need any climbing. And then the trait of adoption, they are well documented here. We have this breakthrough product is very important for the breeding program to know exactly what we are breeding for, which region are we targeting, and then what exactly the end user are looking for. And also we have the essential traits, but the key adoption trait, which means the breakthrough product must have it before we release it to the market. To achieve the breakthrough product and then our breeding objective, what do we have in terms of genomic resources? So since my work span generally on genomics aspect, I will focus more of the presentation on the genomics. So how are we using the genomic resources we have generated so far to improve the yam breeding efficiency? So for that, we have genotype 300, we have resequencing 300 genotype, which include some market variety, land resist, and then improved variety. And then we are using those ones to develop the iterative group and use those information also for some trait association in the breeding program. We have developed a quality control students markers, which is a tool that we are using in the breeding program also for routine activities. We develop how we are using this one and then how we develop this in the breeding program. We have also breeding population that we are using for trait association. And then we have sequenced 35 genotype using the PacBio combined with high fidelity, and then we are using it for pangenome. And we have 3,000 SNPs marker used for mid-density data, which is very useful for our product advancement. And then we recently sequenced more than 900 YAM genotype for Illumina for around 30 years. And then we are doing some expression analysis also. And then for germplasm management, you can see here now what we have done. We have our germplasm, we have a pipeline in place already. And then we are using this one to monitor the diversity we have. And then for monitoring our diversity, we have generated our iterative group here, what we are using now to kind of develop our population and then evaluate them to make sure that what we have is enough to address the breakthrough product we are looking for. And then for the trait discovery and deployment in the breeding program, we are using here two methods. We focus on the linkage mapping, the association mapping, whereby we have some breeding population historical and then validation process. For the linkage, we are focusing on QTL analysis, whereby we have some crossing made and then we do different analysis to be able to identify the QTL data associated to the trait we are looking for. And then I try here to summarize our trait discovery stage gate. So we have our stage 1 to stage 7. The stage 1 is kind of like the product scope, whereby we define in the breeding program the trait we are looking for. And then do we have the germplasm that we can use for those one, which generally for market survey, all those things. The stage 2, we have the phenotyping donor, whereby we assemble the material, and then we proceed with analysis with the locus identification. and so on until we validate the product. And then we have our trait here. All these traits, they were sourced from adoption trait from our breakthrough products. And then based on the stage gate where we are, we now map all the different traits across the trait discovery stage gate. And then you can see some of the traits they have moved ahead, and then we have validated some of them in the breeding program, while some of them are still at the locus identification, and then they will be moving across the trait discovery and deployment stages. For the GWAS, here we focus on some traits. This is an agronomic to the disease, and then we have identified some very good chromosomic region that are responsive to the disease and then to the tuber yield per plant. And then we have more information on this one. For more detail, you can visit this our paper to see how we proceed with all the analysis, and then to select the different putative gene that we have identified to be associated to the targeted trait we are looking for. A key thing to the yam is the plant cells. And then, as you know, in the breeding program, at the early generation, you can not have the plant cells. So we did some very nice study whereby we focus on assembling some historical data. We use them to detect the region associated to the plant cells. And then you can see here we identify very good that is related to the plant cells, and then we validate them by identifying how those markers segregate in the population we use. And then from here now, we can see that directly in Dialata, we have 92% of the population which are homozygote, which means in Alata we have the ZZ related to the female plant, while the ZW is related to the male plant, which is the opposite in European data. And then I will explain that one later. We have more information on this paper for more detail. And then in the introduction part, I was saying that the heterozygosity in yam is very high, and which means this could have an effect on the heterozygosity analysis on the positive, is it negative? So we conducted a study in the in the breeding program, whereby we define different markers with different heterozygosity level, and then we use them as a covariate in the analysis. And then you can see here, if you select a marker with low heterozygosity, you will not be able to classify your population. Why marker with high heterozygosity, you'll be able to classify very well. And then from this now, we now say, okay, based on this information, having some marker with high heterozygosity could have a positive impact on the model we are using for the detection of QTL for yam. And then we have done that one, and then you can see here, marker with high heterozygosity try to improve the QTL detection, and then you can see here on the QQ plot the observed versus the obtained, which kind of like really improve our model we use. And then here we provided more information, how we selected those markers, how the analysis was done. Everything is documented in this paper. But as takeaway message here, marker with high heterozygosity, if it's included in GWAS model, it improves the detection accuracy. We now move ahead, and then those marker that we select through those marker, we run some heat map, putting the yield in, and then you can see here we're able to classify our population into two clusters. These are kind of like high yield, while this one they are kind of like genotype with low yield. So with this marker, we can segregate very easily our population, which is also a way to kind of like a way to select some genotype in the breeding program. Moving forward to the QTL detection, also we have done a very good job in the yam breeding program, whereby we select parent for the trait we are looking for. This is the case of the yam anthracnose disease, whereby we select the parent, we sequence the material, Then we did some quality test analysis, and then with this one, we can easily predict the diploid level of what we have. We reconstruct the pairwise recombination, and then we got a very perfect recombination, except on few chromosome which did not which allow us to, of course, to perform the downstream analysis. Then for the yam anthracnose disease, we did some detached leaf assay, and then we were able to identify different QTL on three chromosome 7, 15, and 18. And then you can see them, we're able to map them here. And then those QTL, we identify some very good genes that are kind of already identified to play some role in disease in plant defense mechanism. So we are in process to validate all this marker and then also to be deployed in the breeding program. More information is available in this paper also for future information. And then what we are doing also, as I was saying, the tuber quality also have a very good importance for the breeding program. And then, the color, of course, is good to have high throughput information. So currently we are working with our collaborator, France Sirad, whereby we are deploying a high throughput phenotyping that we can use to detect the color. And then from that, use those information now to run QTN analysis, and then we have succeeded for this one to construct our genetics map currently, and then we'll be using this one now for downstream analysis. In terms of marker deployment in the breeding program, those markers that we identified either through QTL mapping and trait association. To validate them, of course, we identify several markers, but not all of them are useful in the breeding program. So we run different analysis, the gene association profiling, and then with some correlation, we come up with this. And then for this, we try to validate some of the markers that are associated to plant cells, to different disease, through a simple regression analysis. And then you can see this marker using 10 different approach. You can see that we have an accuracy range from 72 to 93, which is very, very high accuracy for the breeding program. And then from here also, we're able now to identify all the heterozygosity marker are linked to female plants, which we can easily identify the sex plant at early generation with high accuracy in the yam breeding now. So all this information are already deployed in the breeding program. For the case of the plant disease, you can see here, this is kind of highly susceptible plant with very, very severe disease. This is the clean material, and then this is intermediate. With this one also, we have already identified the allelic information that are associated with low disease. And then with this one also, we have deployed in the breeding program. And then we have two markers, and then all of them, they are predicting into the same positive direction with almost more than 80% accuracy, which is very good for the breeding program. Now, for the QC marker, it's very useful tool to have such tool in the breeding program, whereby you can refer to these tools to conduct some routine activity, species identification, duplicate identification within species, variety tracking. So this tool we have deployed in the breeding program. And then to do this one, we use two methods. We have one method we call the purity, and then the annealing. And then you can see here with 50 markers, for each of them, you can predict almost 80%. Then for this now, we now say, okay, let's select at least 100 markers, which give us almost 100% accuracy. And then with this one, we validate them, and then you can see using a set of 50 markers, you can identify all the yam species. Here we implemented 6 different yam species, which allow the marker can identify them very easily. And then we also identify some duplicate in the breeding program. More information is available in this paper also that we recently published. And then this is how we are using also this marker in the breeding program. You can see here we have the parent, we cross this one, and then from this we can identify very easily the non-hybrid and then discard them at early generation. We are also using this marker to also reconstruct the pedigree of unknown parents in the breeding program. Just sequence them, you run a prediction model, and then with that one you can know which of the parents are contributing more in the to the progeny, and then with that you can reconstruct back the pedigree information. And moving forward, so I will say that we talk about the mid-density SNP markers, which is kind of like cost reduction. So it's very effective to have mid-density SNP markers that you can use to do some genomic selection and then product advancement in the breeding program. So for this, we select highly informative SNP markers, which is around the 3,000 SNP markers well distributed across the 20 yam genome. And then how are we using this marker in the breeding program? The classical breeding program, you can see the number of years that we can use before you release a variety, a total of almost 10 to 11 years. And then from stage 1 to stage 2, we can spend 4 years before we can start recycling our parent here. But using these SNP markers that we develop alongside with what we call rapid seed system multiplication at the third year's application of GPCP, which stands for genomic performance, genomic prediction of cross performance, we can select the best parent and move them back now for crossing, which means with using this method we implemented, we can reduce the breeding cycle from 4 to 3 at least for parental recycling, which is a very good achievement for yam, knowing the lengthy breeding cycle of yam. So another thing also we recently did is the implementation of the GPCP, as I was saying, which is another form of genomic prediction. But using that method, you can see we give advantage to one of the parents to cross for all the potential parents, which is not really balanced in the breeding program. To solve that one, we implement also the GPCP combined with the OCS. The OCS stands for the optimal cross selection, which allow us now to balance the population and maintain the diversity within the population we are analyzing, which is a very good approach for a crop like yam. Now, very recent achievement in the yam breeding program, and then we are very kind of like leading this one in terms of kind of like innovative approach, and also especially for in Africa. We started with the pangenome development for yam. So why do we decide to do the pangenome? We have reference genome already, but the first reference genome is based on single species, and then as I was saying before, we have different yam species, which means you cannot use one species during the alignment, so you may end up with a lot of missing information. The alignment accuracy will be very poor. And then to do that one, we select a lot of yam variety and then yam species, and then we put them to be able to have a common reference genome across all the different yam species. And then we have selected some parents that have good parental contribution in the breeding program. Our first result in the pan genome here now, this is just for one sample data. We try to estimate what we have. And then for the genome scope profile, you can see that we easily estimate the yam genome size to be around 600 MB, which align with what we are expecting. You can see the ploidy level is diploid, which is confirm also what the sample we selected at initial. The error rate is very low, which gives us a very good confidence of our genome sequencing for mission. And then you can see that we have 54 percentage of the yam genome which is duplicated, which is very huge in terms of duplication genome. Then this is kind of like information covering the different species we have sequenced recently using the PacBio combining with the high fidelity. So you can see the number of genotype per species. And then this where we have the AB cross with this is one hybrid that we obtained between Abyssinica and Princelis, which is one of the progenitor of Rotundata. The reference genome is a bit higher for Rotundata, which is of course because it's an hybrid of two species. And then the sequencing information here now is around the highest is within this, and then we have from 90 to 92, which is also very high considering the monocot complexity of the species we are dealing with. And then based on haplotype base here now, we are able to assemble our reference genome into the 40 chromosome. Why do I say 40? Of course, YAM have 20 chromosome, but since here we are talking of haplotype base, each of the chromosome have a 2 haplotype, and then we successfully assemble them into the 40, which is very good achievement for the YAM breeding. What's kind of also very good result we got is we observe some kind like structural variation on chromosome 9, 11, and then chromosome 20 here. And then we explore very well this region, and then you can see on chromosome 20 here, you can see we have some inversion here. And then this inversion could be linked to plant sex. And then here now you can see that we have some missing information here, which could be linked directly to the ZW information that observed in the female plant. So this information, we are now embedded them to develop some microRNA that could be used to kind of develop a new set of markers for the yam breeding, especially for the plant sex. We have also successfully constructed our pangenome based on the kernel analysis, and then here you can see we have a group of Rotundata here, and then we have the second group of Alata here. But what we are looking for here now is this haplotype. Now we are looking at how the haplotype were kind of like assembled, and then you can see that we have some misclassification here with this 194, and then here. But if you look at them, we don't, of course, they are not into the same block, but the correlation, of course, is not so far. So we can still be as accepted. We did some comparative analysis within a genome and then among genome, and then you can see within Alata, the two species, you can see a perfect alignment. Of course, it's expected. This is Alata. But when you compare Alata and Rotundata, you can see you have some translocation and then some inversion across the genome from one species to another one. So these are the two species. This is Rotundata and then this is Alata here. We have some high conservative region across the 20 chromosome. Around the percentage is around 40%. But of course, you can see some part of the chromosome which are kind of like translocating from one region to another one. Chromosome 7 in Alata, some part is equivalence of chromosome 12. And then you can see also the same information here, which need to be kind of like taken into consideration when we are developing a marker. So of course, for here, we prefer to have some very good conservative region whereby we can use those ones for marker development. And then here now, if you look at our pangenome here, these are the reference genome we have sequenced, and then you can see the gap we are having across all the reference genome. That is why now to having this consensus reference genome is a very good idea, whereby we can use this information to do any alignment from one species to another one, which is also a very good achievement. We are adding more variety, more species to improve this information we have for the yam reference genome. We are also doing some functional genomic recently to improve our trait discovery. And then here we are focusing on the clambing, which we have said before is kind of like demanding and then costly. And moving forward, also, we are focusing on how can we modify the clambing habit of yam, and then we are doing some expression. We are focusing also on the tuber bulky. These are the genotype we selected, and then we have the guagua, which is kind of like the semi-dwarf, and then this is our control. Some of those varieties has been released, and then modifying them will be really good for farmer to address the issue of kind of like providing the stake for the yam. The expression analysis, we focus on tuber, stem, flower, and leaves. And then you can see using the expression analysis, we're able to have a very good classification of the tissue, of course, with very little error here. We observe more than a lot of genes, around 5,000 genes, and then we classify them into the three components, whereby we have the biological process, the cellular component, and then the molecular function. We got a lot of more of the genes that are into the biological process, which is very good for us, and then they are regulated. So it's also a very good information for us, whereby we can decide if we are modifying the tuber bulking, it should be kind of like up-regulated in the tuber and then probably down-regulated in the leaves to avoid some contradiction. We now compare all the different genes we observe from leaf for the different tissue we are targeting. And then based on this, you can see we have the highest difference between the leaves and tuber. Of course, those two tissues, they are totally different, and then which can be seen in the number of the genes we are having. And then we run the volcano plot analysis and then seem to the number of the gene up-regulated are more higher than the down-regulated, and it's good for the trait we are targeting, and then alongside with the tissue also. We have identified some very good gene, and then we compare this gene with our prediction, and then we are observing almost 80% of accuracy, which is already very high, doing some normal annotation with expression and analysis. We are focusing on some genes that are kind of like involved on the on tuber bulking, and then this is one of the genes we are targeting. And then we look at the pathway synthesis, and then we have identified some modification alongside the pathway, and then how this one could be also be useful in the breeding program. To do that one, we selected some genes that are already well known in tuber, in clambing and tuber bulking. These are kind of like the reference genes that are well known, especially in potato. And then we run some blast analysis to be able to detect those genes in the rotundata reference genome. And then we get this gene, which is one of the genes that is available in Rotundata already. And then we have designed already the marker, and then we are in the process of validating them and deploy them into the breeding program. In terms of high throughput phenotyping also, we have started this one already, and then we are using the drone to have some image analysis, and then link this one with the tuber bulking, and also to detect if we can use this one to detect the yield at harvesting. We are also using a lot of high throughput analysis for tuber flesh color, as I said it before, and also some high spread analysis for the needs, and then the starch quantity, starch quantification. So all this one has been developed and then been applying the yam breeding program. Using all this technology, we have recently, I think last month, we are happy to release some very good yam variety. This is one of the yam variety I was holding, which is a very good. This variety is very, very dear to us because for exportation, farmer are not looking for, as a farmer, the trader are not looking for big yam for exportation, but yam very compact that easily be packed and then sent. They have very, very good yield advantage, and also tuber quality is very, very good. You can see the flesh color here. In terms of kind of like excellency in the yam breeding program, we have received, the yam breeding has received some award recently, which is very good encouragement. That's mean the work we are doing at least has been recognized. So in terms of, we received 3 awards in India last year, and also received some award in US, especially on the LGBT. Yeah, on the, yeah. Our future plan, we want to extend our collaboration thanks to Carlos, who I can call him now ambassador of YAM in US. He's really, really doing a lot of for YAM to make sure that the YAM is well represented in US. We appreciate you for that. We are still exploring a lot of kind of our breeding population from cultivated to the wide related species to do a some to identify some genotype that so we know that have the trait of resistant to disease. Also, we are focusing on developing some kind like non-staking yam, semi-bushy that could be used to reduce the yam cost production. And then, of course, we are still working on deploying more marker into the breeding program, especially for the food quality. The flowering manipulation, we are happy to have a grant now with the North Carolina, and then I think very soon we start getting a lot of results, and then how we can manipulate the yam to especially for the flower to start getting some very good crossing at early generation. The tuber shape index and then the tuber flesh color, we are working on that one, and then we'll give more information as soon as possible. The pond genome, we deploy this one now into the breeding program alongside with the functional genomics, and the nematode also will work on it. And then we are also open to kind of start doing some phenotyping on the droughts, and then exploring of genomics of wide related to identification of useful allele for yam improvement. So I think with that, I will. This is my last slide, and then thanks to the YAM team, both in Abuja and then in Ibadan, and then our national partners across Africa. And then also we have some good collaboration in Japan and BRC. And then in the US, we have now the North Carolina State University, Alpha Houston. And then we have the Salk Institute. We have Cornell University. So the work I present is representing what we are doing, and then how best we are to do our work to make sure that we deploy innovative approach in the yam breeding. So with that, thanks so much. And
Thank you.
yeah.
Thank you, Agre. I want to stop sharing so we can open the. Please post your questions in the chat for Agre. One of the questions I have is I know you have listed there yams as diploids, but has anybody observed variation in chromosome number and ploidy level chromosome number within species? Because this type of propagation and reproduction, vegetative propagation, plus the fact that sometimes some of the materials are very difficult to get them flower, hints at potential for variations in chromosome number. Has anything been detected?
Yeah. Within spaces to the last of my knowledge now, we haven't seen different ploidy level, especially in Rotundata. But in Alata, we have seen some kind of little variation, especially we have seen some diploid and then some tetraploid, and then
Mm.
yeah, but within Rotundata, we haven't seen any variation yet. A key recent study that we conducted, but we want to validate it now with the pangenome to see the ploidy level of the interspecific progeny we got within Abyssinica and then the Princilis to be able to confirm what we saw during the chromosome counting. So we have more information when I complete the pangenome analysis, so I can provide more detail on how the how the ploidy vary within species and then among species.
Okay. Question from Roberto here, as he states, how do you model genomic selection with plants that have different sexes, males and females?
Yeah, so generally for this one, what we do, we don't consider the sex at initial when we are running the analysis. So we run the genomic selection without the sex information. And then once we select the parent now, we use the sex to be able to make our final selection. But what we did with the Cornell University with the GPCP approach, we factor in also the sex information. So with that sex, we make sure that during the analysis pipeline, we don't select only the female or the male to end up with not be able to make the crossing. So we have add that one in the pipeline.
Mm-hmm. Okay. Any other questions from the audience? Um, I I mean, what you're doing is key to the future of yams. I do think that there's a big need for a overhaul in terms of the production system of yams. You listed some of the constraints that are sort of pain points. The vining and the need for trellises is one. The other one is the need for, I understand that up to a third of the crop is saved as seed. Okay. So that's a huge cost for a crop that has a high value. Okay. Farmers need to save one third of the crop, up to one third of the crop as seed. So that's another area that probably deserves more focus. And as you rightly pointed, the tuber shape and the potential for developing simple equipments to help in harvesting yams is very important. Okay, if we keep producing yam as it has been produced traditionally, it is a crop that can be endangered, I would say. A bit extreme, but I think we need to think out of the box on how can we help farmers produce yams in a more efficient and productive way. Okay, and what you're doing is key for that, Agre. Okay, all right.
Yeah, thank you, Carlos.
Yeah. Okay, a question here from Omena. How can the integration of pangenomics with high throughput phenomics, phenotyping, and other omics create a more holistic decision support system for selecting superior parents in a yam breeding program?
Okay, yeah, thanks so much. So one, for the pangenome help you want to kind of like capture all these structural variation you have in your population. So using the pangenome, you are eliminating any possible error that you are not going to miss any part of the gene or any part of the information you may have not be able to capture if you use probably some mid-density SNP markers or targeted sequencing information. Say the whole genome help you to capture all these structural variation. That is one. Combining with the high throughput phenotyping is a very, very good approach because we cannot do only a pangenome and then just make the decision. We still need to go back now to do some high throughput phenotyping, especially in terms of kind like the climate change we are looking now. So combining with high throughput phenotyping in terms of kind like the drought, whereby we are focusing now, can help you also to identify those key genes that are playing a very a good role in terms of understanding uh, allelics related to a climate. So that is one of the aspect of the high throughput phenotyping. For the omics, it's very important to include omics approaches because for the omics, you are dealing directly with the expression with the RNA. And then here we have removed all the environmental aspect, which means anything we are seeing that is expressed is true definition of the plant behavior. So all of them, they work in synergy to be able to make a final decision for the yam breeding. So the feature is there combined, but of course is is a bit expensive. But of course, the pangenomic doesn't work alone. It has to go alongside with the high throughput phenotyping, and then with also with the omics of course.
Question that came to mind, and you may have mentioned this, but how much use have you making of comparative genomics with other monocot species that are more developed or that have more
Sorry, Carlos, I did not get your point.
more comparative genomics with other monocot species that are more
Yes.
advanced? Yeah.
Yes. Sorry, I did not present that part in there. We did a lot of kind of like comparative genomics within the monocot. We compare it with maize, with dates, with Elias guinensis. And then, yes, surprisingly for us, the yam is very close to Elias guinensis. So I'm still working on,
Really?
yes, to the palm tree.
All
So
palm,
yes. So not only my study, I think when I was doing literature review, also I have seen that one in some paper. So I'm still kind of like checking more to see what really happened. Of course, all of them they are monocots, but one is climbing and then another one is a tree. So yeah, yes. So we
Okay.
have done a lot of comparative analysis with monocot.
Okay. This is a clarification question from Deepa in the chat. It says, the pangenome results shown here, are there from 300 diverse genomic sessions? It's a question.
No. We don't have such a resources to do pangenome for 300. It's really expensive. So what we did here from the 300, we narrowed it down to 178. So those 178, they are unique population. So and then from those 188 now, we now select a representative of each group. So we end up with 35. We selected the 46 first. But after sequencing, we have some genotypes that are not good, and then we discarded them, and then we end up with 35. So the pangenome I presented represent 35, and then we are adding more. But that's everything I presented today is around the 35.
That's good. Any other questions before we I apologize again for the hiccups that we had at the beginning. Somehow I was kicked out of Zoom and I was not able to get back for a while. Anyway, okay, can you see my screen there? I imagine you can, isn't it? So that was the introductory screen. One of the things that talking to Agre before we started the seminar that really surprised me is that Agre is from Benin, Republic of Benin, and he's actually from the same region, the same village that Matthew Ayanan, the speaker for the second seminar in this series, is from. That's very interesting. Okay. Next Thursday, we're going to have Julia Sevilla from CIMIT. I think she's based in Zambia, I understand, or maybe wrong, but talking about food system transformation in Africa and the relevance of biodiversity in vegetables. Thank you so much, Agre, for your presentation, and thank you for all the work that you're doing for yams, okay? Thank you, Brandon, for your help. You really saved the day today with my hiccup. And thank you for all of you that attended. We look forward to have you next Thursday for our fifth seminar of the series, okay? You guys have a great weekend and see you next week, okay? Thank you. Bye-bye.
Okay. Thank you. Bye.